Hey AP Psych students, it is Megan with Fiveable. Tonight we are covering the biological basis of behavior, specifically neurons and neurotransmitters. My goal is to help you guys prepare as best as possible for the upcoming AP test in May. And so if you have any questions or anything tonight that you want to know more about, feel free to either put them in the chat, or if you have a really specific question that everyone would benefit from, on Crowdcast, there's a tab at the bottom of your screen that says Ask a Question. You can just click right on that, type in your question. It will send me a notification, and I will answer it at some point in our broadcast tonight. So that's how you can kind of interact with me. Later, I'll also be posting some multiple choice questions so you guys can go in the polls and actually answer those as well. So that way you can interact with me as well. So getting started then for tonight, looking ahead at some of our review sessions, Tuesday next week, and typically we are on Tuesdays. Unfortunately, this Tuesday I had something come up, so I can't broadcast, but typically we'll be on Tuesdays. Next week, we're covering more of the brain and the body systems, and then we move into states of consciousness with dreams and drugs, which is a pretty small unit, so we're just going to do one session on states of consciousness. And then we get into another heavy hitter, which is one of my favorite units, sensation and perception. We'll talk about various thresholds and detection of signal and stimuli. So that's where we're headed. As for today then, we are in the biological basis of behavior unit. The focus of this unit is kind of the physiological processes, the things that are happening inside my brain and inside my body that are creating my behavior, my actions, those sorts of things. This is a heavy hitter unit. It's going to cover 8 to 10% of the AP exam. And not only that, but so much of what we do in this unit keeps coming back over and over and over again. So really doing a good job on this unit is going to help you so much down the road. And so I would recommend, even if you've already tested on this unit, just reviewing some of the big concepts is going to be really helpful to you. Specifically, this unit covers all the different parts of the brain, the nervous system, specifically neurons, and specific neurotransmitters as well. So there's lots of stuff wrapped up into this unit, and we're going to do our best to kind of break it down and make it less confusing and less challenging for you. So tonight, our focus is neurons and neurotransmitters. And so the first thing that you guys have to know in this unit is you just have to know the parts of a neuron, and you need to know how it communicates inside itself. And so this diagram here, you've probably seen one similar in your book, but you have to know all of these parts. So you need to know that the dendrites there, those spiky little things, are what receive the messages. They kind of suck them inward into the cell body. And then that message inside the neuron, this is all one neuron we're looking at, travels down the axon. And another thing that you need to know is the axon is sometimes covered in a myelin sheath. Not always, but you need to know that a myelin sheath is like a coating that's kind of wrapped around the axon of the neural fibers, and that helps speed up the impulses. So it allows us to send messages faster. And actually, when people develop diseases like Alzheimer's, it's believed that the myelin sheaths are deteriorating, and so their messages don't send as smoothly. So that's sometimes believed to be what's going on. So myelin sheaths are important. We like them. They help us communicate faster. So the message is transmitted down the axon. That's considered an electrical impulse. So that's sometimes, I don't know how specific they get. I have seen it mentioned though, so knowing that it's an electrical, not a chemical impulse is important. So the axon shoots the message down to those other little branchy looking things at the end, and those are called our terminal branches. And that's the end of the specific neuron. And at the end of the neuron, you see those little kind of balls at the end of the terminal branches. Those are where the neurotransmitters are kept, and they shoot out from there once the message is received. And that's how neurons communicate along the line. So just kind of knowing, I've seen questions before on the test where they say, hey, what is the correct order that a message travels within a neuron? And it'll list all the parts of the neuron. So you need to know that it goes dendrites, then cell body, then axon, and then terminal branches. So there's really no super creative tips or tricks to remember that. One thing I would suggest, and I have my students do this, is we actually build neurons We build ours out of candy, and we label all the parts. Giving yourself some sort of context for remembering all the different parts, I think, would be really useful. So we use like the cell body is a marshmallow, the dendrites are pretzels, the myelin sheath or the axon is like a Twizzler. And so they make the neuron out of all of these pieces of candy and label it. And then later on, when they're trying to remember the different parts of the neuron, it helps to like, oh yeah, we used pretzels. What were that? What were those things? And then it kind of helps jog your memory. So if you could come up with some creative way to build a neuron, I've seen people use like pipe cleaners and buttons and things like that. That might also be really useful to you, giving yourself some context for remembering what is what instead of just memorizing these kind of weird words that you don't know. So that's how a message travels inside the neuron. And we're going to look a little more specifically at the action potential on the next slide. So the action potential, what you're looking at right here, is occurring in the axon of the neuron. And this can be kind of confusing and kind of tricky. So what happens is when a message is received, if that message is strong enough, it continues on and triggers what's called action potential. If the incoming message isn't strong enough, nothing happens. Neurons don't like slightly fire or half fire. They either fire or they don't, and that's called an all or none response. So if that incoming message is strong enough to trigger a response, the axon starts to get to work. And so if you look, there's those like blown up tubes there that are showing us maybe what an axon would look like. And so the inside of the axon is made up of mostly negative ions. And what happens is as action potential kicks in, the fluid interior starts to switch inside the axon. And so sodium ions, which are on the outside of the axon, rush in, and then they push out potassium ions. And I would try to remember sodium and potassium. Sometimes they do get that specific. So sodium ions are rushing in, and then the potassium ions are pushed out. So it's kind of like this, like they're in and out, in and out, in and out. And this pushing in and out that those ions are doing is what's pushing the message down the axon. So sodium goes in, potassium out, sodium in, potassium out. And as we go down the axon, that's what's pushing the message along. Obviously, it happens a lot quicker than this little slow wave that I'm doing, but that's kind of the gist of how the message travels inside the axon, and you do need to know that. I don't think you need to know too much more specifics besides that, but just understanding that basically action potential is causing that inner negativity of the axon to depolarize, and then basically action potential is the axon's way of repolarizing itself, kind of coming back to its neutral state. So those ions are fluctuating in and out until it returns to a normal state within the axon. And that's how the message is pushed along. That also means that we can't just continuously send a message down the axon because we have to wait for it to repolarize before a new message can come along. And we're talking about it like this is a slow process that we have to wait, seconds for. But in reality, it's like, it's happening super duper fast. But we still have to wait for it to happen before it can fire again. And that's called a refractory period. So neurons have a refractory period. They have to repolarize themselves before they can send another message again, send another message, send another message, so on and so forth down the line. Okay, so that's within the neuron. We looked at the big picture, all of the parts of the neuron, and then now we've looked inside the axon at how all of those little specific things are happening. So if you have questions about that, throw it into the question box. I will answer those, but otherwise we're going to look at now outside of the neuron, how that communication kind of continues onward after it leaves the neuron. So communication maybe seems a little simpler. I think it's a little simpler. So we get to the end of the neuron, and that's where those terminal branches are, and you saw those little tiny buttons on the terminal branches, and that's where the neurotransmitters are kept. And so what happens is those synaptic vesicles, that's what we call them. You might also see them called terminal buttons. And that's the kind of tricky part. Sometimes they use different words for the same thing. But those little balls at the end of the terminal branches release neurotransmitters. And those neurotransmitters cross this tiny gap called the synaptic gap to the next neuron. And that's one thing I can't stress enough. Neurons do not touch. Never, ever, ever do they touch. So you have to know that. So there's always like a tiny gap. It's called the synaptic gap or the synaptic cleft. So the terminal branches of neuron 1 are shooting out these little balls. You can see them in the image here, the little neurotransmitters. And then those neurotransmitters are trying to find the next neuron to cling to. And so we have the dendrites then over here of neuron number 2, and these little balls are floating around, and they try to find the dendrites of neuron number 2, and then they suck in the message, and then the process repeats itself all the way down the neuron, and that's how they connect to one another. Not all of the neurotransmitters, all these little balls floating around, they don't always find a home on the next neuron, and so what happens is they get sucked back up into the sending neuron, and that is called reuptake. If not all of the neurotransmitters find a home, they are sucked back up into the original neuron, and they remain there. So that's a little bit easier. Basically, neurotransmitters shoot out, they find a home in the dendrites of the next neuron. Let's see. I think that is about it for the communication between neurons. We can also impact how long the neurotransmitters stay around. If you have, like if you take SSRIs, which are selective serotonin reuptake inhibitors, like someone who maybe has anxiety or depression, you might take drugs called reuptake inhibitors, and that's literally what they do, and you'll learn more about that later. But basically, I'm taking this medicine to help with my depression, and what it's doing is it's preventing that neurotransmitter of serotonin from getting sucked back up. It makes it stay around longer and gives it more of a chance to cling to the next neuron and then move on down the line, because we want that serotonin. The serotonin makes us feel good. It lifts our mood. And so by taking a drug, it can actually alter how the neurotransmitters are received in our body. And that's actually how a lot of medications work. So you'll get a better understanding if you're on any sort of SSRIs or things like that, how they actually work in your body. So, all right. Specifically then, I like this analogy of neurons firing are like flushing toilets. You might not think so, but after we look at this analogy, it's going to make sense, and it's going to really help you remember all of the parts of how a neuron fires. So first, if you think about a toilet and you just jiggle the handle, you don't push it all the way around, but you just shake the handle, nothing happens, right? Your toilet's not going to flush. It just sits there. It does nothing. Just like a neuron, if you don't give it enough to reach that threshold, nothing happens. And so if you do push enough pressure onto the handle of the toilet, it flushes completely, not partially, which is our all or none response. So if you reach that threshold of pushing on the lever of your toilet, you can create the all or none response, which is the toilet flushing or the neuron firing. If you think about what goes in a toilet, those gross negative contents, the negatively charged contents, wash away. Just like as you look at down the axon, the negative depolarization happens in the neuron. It gets flushed away. That's how the message sends, just like in a toilet. Luckily for us and for neurons, they only fire in one direction. Your toilet can't go backwards and shoot the water up at you, hopefully, unless there's a problem. And your neuron can't backtrack and send a message backwards. The message only flows one way in a neuron. And then lastly, if you flush your toilet, you can't immediately flush it again. You have to wait a few seconds or maybe a minute, however long. Just like that with our neuron. You can't immediately fire your neuron again. You have to wait for it to finish running before it will go again. So just like with the toilet, you have to let the water finish flowing before you can push it to flush again. So that is my toilet analogy for how neurons fire just like we flush a toilet. So hopefully you find that useful for remembering specifically how a neuron fires and the different parts of the neuron. And so I mentioned before an electrical impulse versus a chemical impulse. You do need to know, at least on the surface level, the difference between those when we are firing our neurons. Some neurons are electrical all the way through, and electrical is a little more efficient. It happens constantly, it's quick, and so we use electrical impulses in our bodies for things that always have to be happening, like our heartbeat, right? We need a heartbeat. We can't just take a break from that. And so that would be an electrical impulse from our neurons firing. Same thing like breathing, just any kind of basic bodily function that needs to be happening. Those are going to be our electrical impulses, how the neurons fire. They're quick, they're efficient, and they're constant. Chemical synapses are a little bit different. So this is where the gap between the neurons, remember, neurons don't touch, the gap between the neurons where information is transmitted chemically through neurotransmitters. So neurotransmitters are chemicals, therefore that's a chemical synapse or a chemical reaction that's happening. Inside every neuron, doesn't matter what kind, it's an electrical message, but then the message that's going across the synaptic gap can be either electrical or chemical. And so when we're talking neurotransmitters, we're talking chemical. We have many more chemical synapses in our bodies because they're more precise, like lifting my finger is one, turning my head is another. All of these different things that I'm doing, the movements I'm making with my body, all of those are precise, refined functions that come from chemical synapses. They're also more selective. So they're more specific about how and when they fire. So electrical is quick and efficient, chemical is precise and selective. So just make sure you kind of know the difference between those two. I don't think they're ever going to get nitpicky and ask you a specific type, but just kind of understanding the difference between those two big ones I think is useful. So the other two things that I wanted to denote the difference between would be sensory and motor neurons. And we'll talk next week. I've got this whole dance that we're going to do about body systems. And so you're going to learn to dance with me a little bit. But we're going to do a dance about body systems and we'll learn more about these neurons. But specifically speaking, we also have sensory neurons and motor neurons. And I think if you think about what each of those words means, it will make sense for you. So our senses, like let's use touch, for example. Our senses, like my fingers, take in information. So sensory neurons carry messages inward into the central nervous system. And the central nervous system is like my spinal cord and my brain. That's where all the processing happens. So when I touch something, when I smell something, when I taste something, my sensory neurons carry that info inward. It's an input thing. And so then they're processed in the brain and then the motor part happens. But for now, let's just focus on the sensory. So that input message is coming in to be processed. And how I remember that, because afferent and efferent are very similar words, is that A for afferent starts the alphabet, and A comes first because you need input before you can have output. So because A comes first in the alphabet, we need the input from the sensory or afferent neurons first. So maybe that will help you remember which is which when it comes time to take your test. And those terms I've seen used pretty interchangeably, sensory or afferent. So you just kind of have to know which is which. Then the motor neurons or the efferent neurons carry the messages outward from my central nervous system. And so that is an output message. It's my reaction to basically the sensory input that's come in. And so to remember that, I think about the word efferent, kind of sounds like effort. You have to put in a lot of strong effort to do something, just like it takes effort to move my arm and effort to turn my head. It takes effort from the efferent neurons. That's kind of how I remember that, just as like a little tip or trick for you. So that's about it for specifically neurons. I'm going to move into neurotransmitters in a second, but I kind of want to pause. We haven't really had any interaction yet. Is there anything anybody wants clarified or wants me to talk further about? Because sometimes these things are confusing, especially if you don't have a strong like science background. This is all kind of new to you. But these are important because these are kind of the basis for a lot of what's going to be happening later on in the unit. So feel free to throw something in the chat or to throw a question up in the little section there at the bottom of your screen. I'll kind of move on, and I'll just keep my eye out for any questions because I'm hoping you guys have something to say. Otherwise, be ready to answer some multiple choice questions in a little bit. We'll get that going. So neurotransmitters, now we need to look more specifically at what those are. So remember, those are the little balls that are released from the terminal branches of the sending neuron. They're the messenger that goes across the synaptic gap. So neurotransmitters are chemical. I just say that. I'm trying to reinforce that. Chemical messengers that transmit the signal across the synaptic gap from one neuron to the other. Because remember, there's no touching. The neurons don't touch each other. There's always a teeny tiny little gap that exists there. And so those little messengers are shooting across the gap, trying to reach the next neuron and continue the message along. There's over 100 different types of neurotransmitters in our body. For the purposes of this class, you probably need to know about 6 of them. We'll talk about the 6 you need to know a little bit later, but just kind of knowing that there's more than just those 6 big ones is important. And each one is super unique. And so what's cool about neurotransmitters is they don't just bind to any neuron. They have like specific homes that they're looking for. And so it's like a lock and a key almost. They fit into a certain receptor site, which then allows that message to continue onward. They don't just get sucked into any little spot. They look for a home. It's like a lock and a key. They fit together. And so depending on how many of those neurotransmitters hit determines whether or not the next neuron fires. Because remember, we talked about this. A neuron doesn't half fire or partially fire. It either fires or it doesn't. And so if we get enough of those neurotransmitters to bind onto the next neuron, then we can send the message along. But if we don't, then that message doesn't get sent and it stops wherever it is going. So keep that in mind, the all or none response. And so just some things about neurotransmitters, because you're going to also see some things called agonists and antagonists, and I want you to be able to differentiate between them. So neurotransmitters come from within us. Our body naturally makes these chemicals. And they can both excite or inhibit neural communication. So we have neurotransmitters that will excite, meaning they want to keep neurons going and firing and going really quickly. And then we also have ones that inhibit neural communication. So they kind of slow it down and they kind of dampen what's happening in our bodies. And again, we'll look at specific ones, but the specific ones that you need to know for the AP test would be acetylcholine, which is sometimes abbreviated as ACH, dopamine, serotonin, norepinephrine, GABA, and glutamate. And I will talk about each one of those a little bit more later. So don't freak out by all of those words that don't seem to make sense right now. But those are specific neurotransmitters that are happening in our body. They're the chemical messengers that are sending, messages throughout our body all day, every day as we go. There's other things called agonists or antagonists that come from outside of our bodies. And those are typically like drugs, as in it could be like an illicit drug like heroin, or it could be like I mentioned before, like a pill that you take that the doctor prescribes you. And so basically agonists and antagonists mimic what's already happening with the chemicals in our body. So agonists bind to the receptor sites. So remember, the neurotransmitters shoot from one neuron and they're trying to find the receptor sites on neuron number 2. Well, agonists do the same thing. They bind to these receptor sites and they mimic their effects. So they do the same thing as those neurotransmitters. Antagonists, on the other hand, they still bind to the receptor sites, but they block neurotransmitters from actually binding on. And so they prevent neurotransmitters from doing what they want to do. And so if you know, like an antagonist in a book is kind of like the bad person who's preventing like the hero from doing what they want to do, then you could think of that's what an antagonist does as well in the body. It blocks the neurotransmitters from doing what they want to do. And so some examples for an agonist would be heroin. So heroin mimics the effects of some of the neurotransmitters in our bodies, which is why heroin makes us feel good, makes us feel relaxed, because it mimics some of the dopamine and stuff like that that's going on in our body. That's also why when you like go through withdrawals from something like heroin, because there was so much of it bound to those receptor sites, your body produces less of those neurotransmitters over time, because it's like, hey, something else is already doing our job for us. We don't need to do it. So when you're coming down from heroin, your body's also not producing enough of that chemical. And that's why you feel so yucky when you go through like withdrawals and detox, because your body stopped producing that. And then you just feel like crap, basically. And that's why withdrawals don't feel very good. Let me see here. Looks like we might have a question. Yep, so someone asked, can we access these slides? My class is not on this neuro topic yet, and I want to access later. Yes, so at the end, we'll talk a little bit about subscribing and getting access to this, but if you do subscribe, you will have access to this, and you can replay it as many times as you want, which I would encourage you to do as we review as well. Good question. Thanks, Yasmina. All right, so that's the difference between neurotransmitters and agonists and antagonists. Now I want to look specifically at the different types of neurotransmitters. For these, I have a few kind of like tips and tricks to remember them, and some of them are just more memorization. So the first one is acetylcholine that you need to know. And acetylcholine is abbreviated ACH, but it enables muscle action. So me being able to move my arms and do any sort of thing with muscle involves ACH. So it's learning and memory as well. So there's two kind of key components: muscles, learning, and memory. And that has to do with acetylcholine. And how I remember it is if you look at the word macho, like macho man, ACH in macho is ACH, acetylcholine. And so it helps me remember like muscles and strong and action and things like that. So that's a hint if you forget what acetylcholine does. Dopamine then is another neurotransmitter, and this involves voluntary movement, but more importantly, and this is the one you really should key in on, is that it involves reward centers and pleasure. And so like I talked about with the heroin, when it mimics the dopamine, it makes you feel really good when you're on it. And so dopamine is all about finding that pleasure, whether it's through food or sex or any other thing that brings us pleasure. That's the dopamine in our brain talking. It's the feel-good centers that brings us that reward feeling. Serotonin, on the other hand, has to do with mood. And so sometimes people confuse dopamine and serotonin, but dopamine is more reward and feeling good, whereas serotonin is your actual like mood or behavior. And so serotonin regulates our sleep, our eating, and our mood. And how I remember mood is that serotonin has two O's and mood has two O's. And so that kind of links those two together. And so when we have depression, a lot of times we can link it to serotonin. And so that's sometimes a question that they might ask on the AP test. They might ask you to talk about what disorder might be linked to one of these 6 neurotransmitters. And so depression would most likely be linked to levels of serotonin. And I mentioned before, if you take a selective serotonin reuptake inhibitor, an SSRI, if you're prescribed that by your doctor, that's actually what it's doing, is it's preventing your body from reuptaking that serotonin. It wants the serotonin to stay around so that you get more of it and feel better, basically. The other 3, norepinephrine, has to do with being alert and aroused. Agonists of norepinephrine create like a euphoric feeling because that's how you feel. So if you like get your heart racing, you feel alert, that's norepinephrine firing. That's kind of like your upper in your body. It makes you alert and aroused and focused and attention, that sort of thing. GABA is an inhibitory neurotransmitter. You should know that. And basically it prevents your body from overstimulating. So it kind of like slows you down. It pumps the brakes for you and prevents you from like getting too wired and too stimulated. So that's an inhibitory one. And then the last one is glutamate. Glutamate is excitatory, so it's the opposite. It wants to amp you up and push you. And it's involved in memory. And then the big thing for glutamate, this is the question I most commonly see, is that it might be linked to migraines and seizures, which kind of makes sense. If it's an excitatory neuron, and think about it, if it's like really firing out of control, that would make sense why you would maybe have like a seizure or a migraine, because there's too much stimulation happening in your brain and in your body, and so it's leading you to experience like a migraine or a seizure. And so glutamate is thought to impact both of those things in the body. All right, any other questions about neurons, neurotransmitters, any of the things we've talked about tonight? Otherwise, we're going to talk about answering multiple choice questions in a second. And so I'm going to set up the poll right now. So if you give me just a second. Question one. So as I set up the poll, basically what I'm going to do on the next slide is show you a multiple choice question. You'll just click on A, B, C, D, or E. It's anonymous, so you don't have to feel embarrassed. If you don't know for sure, at least take a guess as we go. And then just make sure you're answering, because I'm going to put more than one question up. So make sure you're answering the correct number question. All right, so if nobody has any other questions, I'm actually going to stay on this slide for a second because I want you to be listening. So answering multiple choice questions, there's a few things I want to just talk about strategy-wise. First and foremost, a lot of the multiple choice questions on the AP test aren't just going to be like, identify this word. It's not just straight like definitional. You have to know how to apply them. And so sometimes you need a little bit of a strategy moving forward in answering the questions. So there's a few things that I would like for you to just kind of think about and do. First and foremost, I strongly recommend mocking up your test. So if your teacher lets you, just write on your test. So cross off like the wrong answers for sure. This helps you because if you get stuck and you need to come back later and review, you already know that you like X'd off the like obviously wrong answers, and so it saves you some time. Other things that I would do is if it's a long question, sometimes they try to trick you and they put in like extra information. So read for what the question's actually asking, because sometimes they can be a little confusing. And so only pay attention to the information that's relevant. I would cross off all the extraneous extra information. Another thing that I recommend is I would just never skip a question. Even if you don't know, at least guess something, because I've seen it happen before where a kid means to skip like question 12 and then they forget to leave a bubble blank and then their whole test is off because they forgot to skip a bubble. And then all of a sudden, you have 5 minutes left. There's no way you're going to ever go back and bubble everything in time. So I would never, ever, ever skip a question. There's no penalty for getting it wrong. So you might as well just bubble something and then put a star next to it and come back to it later. And then the last just kind of other tip that I have about multiple choice questions is that if you're running short on time, you should bubble all the same letter. So like pick A or B or C. I don't know if that old like adage about picking C is correct or not, but just pick one letter and stick to it. You're more likely to get more of them right if you bubble like the last 10 all C or the last 10 all D than if you like skip around the letters. So if you're running short on time and you're afraid you're not going to get to all of the questions, do yourself a favor and quick answer them all. Maybe you'll get a couple right, and that would be great. So, all right, specifically now, practice question number 1. Like I said, you guys can go into the poll right now and select your answer. I'll give you like 30 seconds to answer it. But in transmitting sensory information to the brain, an electrical signal travels from the blank of a single neuron. So this is asking you to identify how the information moves down the neuron. So go into the poll right now and select your choice if it's A, B, C, D, or E. I'll give everyone a minute to think about that. And again, it's anonymous, so don't feel bad if you don't know. Take a shot at guessing. It's better to at least try than to not try at all. We've only got 4 votes. There's more of you out there. I'm going to give you just a little bit more time before we move on to the next one. And I'm adding some other ones right now because we're going to look at some more questions. Oops, I messed that one up. Hold on 1 second. We're almost to that. C, D. All right, so question 1 is asking basically how the neuron travels. And so the correct answer is D. So for this, basically it's an identification question about the parts of the neuron. And so you have to be able to put the parts of the neuron in order. And so it looks like 90% of us in the poll got it correct, which is great. And these types of questions sometimes I think can be tricky because you see dendrites at the start and you think, oh, dendrites, and then you circle your answer. But sometimes there's more than one that starts out correct. And so you really have to take your time and read these questions and make sure you can identify all the parts in a row. So the dendrites then suck it into the cell body, which then sends the message down the axon. So D is the correct answer. Good job. All right, question 2. The slowdown of neural communication in multiple sclerosis involves degeneration of the, and I think I gave this one away at the beginning. So, but think about what we talked about helps speed up neural communication. And that's question number 2 in the poll. So see if you can answer that one. See some answers coming in. I'll give you just a couple more seconds. I'm just adding more poll questions here so we can keep answering. All right, let's look at what you guys came up with. Question 2. Yep, everyone got it right with E, myelin sheath. And so the myelin sheath, again, we've got the axon, which is like the neural fibers. The myelin sheath wraps around the axon, and it basically is just like a tubing that kind of helps it move faster. Like think about how an electrical wire is sometimes like wrapped with something to help it like, I don't know. I'm not an electrician. I'm assuming that's why they're wrapped. Maybe not. But it wraps around the neural fibers and it helps transmit the message more quickly. And so in multiple sclerosis, that message doesn't move as well, basically what's happening because the myelin sheath has deteriorated. All right, question 3. Within a single neuron, the action potential. So again, we're in the polls here at the bottom there. You can all click on and participate, or you can ask me a question too if something's popped up, because I'm still open to taking your questions. So we're on question 3 in the poll section. See if you can come up with that. I'm posting one more. All right, so it looks like people are putting A so far, and that is correct. Remember the toilet flushing analogy I gave before? Thank goodness a toilet can't shoot its contents backwards, just like an axon can't shoot its message backwards. The message travels one way down an axon to the terminal branches or the axon terminals. Good. Question 4. Psychoactive drugs interfere with normal neural transmission. Where does this interference take place? And we did not specifically talk about this. So if you're not sure, that's okay. We'll talk about it after you guys take a shot at it. So that's question 4. I'll give you like 30 seconds to try and answer that. All right, let's check out what we're coming up with here. Good, everyone's putting D. That is the correct answer. So the synapse, that kind of gap that exists, that's where the psychoactive, sorry, I'm talking too fast. The psychoactive drugs go in there and they interfere with the neurons actually being able to, the neurotransmitters being able to impact the neuron. Good. Last question. A person with schizophrenia may have an overactive dopamine system. Drugs used to treat this disorder prevent the action of dopamine by keeping it from binding to its receptors. These drugs are... Yeah, you guys are rocking it. Most of you put E. That is the correct answer. Antagonist, because remember, the antagonist in a book or in a movie prevents the good guy from doing what it's supposed to do. So drugs that help prevent the dopamine because there's too much of it is an antagonist. It's blocking it. It's preventing it from doing what it's supposed to do. So not all antagonists are bad. That doesn't mean bad necessarily. It just means preventing or blocking it from doing something. I have just a couple other things I want to talk about in regards to review, but that's kind of it for content tonight. Is there any other questions? I really hope as we go throughout the year, we can be a little more interactive. I like when you guys ask questions because sometimes, you know, I might miss something, or maybe your textbook talks about something different and you want clarification on it. So I really I'm going to make these slides that kind of are general, broad overviews, but if you guys have questions or things you want to talk about, I'm totally down to do that as well, because this is for you and your benefit. And so I want that to happen. So a couple of things. These were free sessions. I think I've talked about that before, but starting in October, this service, you do have to subscribe to Fiveable for. And so the link is up there. You can just go to, if you Google Fiveable, it's easy to find. But basically, you do have to join, and there are membership benefits. There's a few things. First and foremost, the earlier you sign up, the cheaper you get it for. So don't wait till May and then, oh crap, I really should go back and review this stuff. If you sign up now, you get the early bird special, and you get all of the services right off the bat. And so it's $15 per subject for the year, or $30 if you're taking 4 APs because you're super into APs. You can subscribe to all of them for $30 a month. What I really like about this is, even if let's say you tested on bio last week and you're like, oh, well, I've already tested on bio. This is still going to be a good review for you. What I have my kids do is every month or every 2 months, we take tests that go back and review bio and research methods or whatever you guys did at the beginning of the year. Because if you wait until May, you're going to have a really hard time remembering the bio that you covered in September and October. And I do see we have a couple questions. I'll get to those in a second. But I really want to stress how important it is, however you're studying for the AP test, that you review kind of consistently. You don't have to spend hours a week or hours a month even, but just going back and refreshing your memory about bio once every month is going to be helpful to you, because when it comes down to May, you've already covered 15, 16 units, and trying to review all of that at once is hard. And so even if we've already covered bio in class, you can always come back and watch it. So in December, you could watch the bio video over. In April, you can watch the bio video. So it gives you access to this consistently, which I think is a benefit. This is literally cheaper than obviously any tutoring service your parents could pay for, or anything that you can get any sort of live interactive thing. And then lastly, last year, the kids who subscribed to Fiveable saved over $1 million in college credit because they reviewed often, they participated in these live sessions, and they earned college credit. And college is crazy expensive. You know that. Your parents will be really grateful if you do well on the AP test and save them some money as well. So let me just check the questions. Chad is asking, will you talk about brain structure or functions in this video? Next time. So on October 2nd, my next session, we will go over all the structures of the brain, all of the body systems, that sort of stuff. So if you are interested in that, then you should subscribe, because that's not going to be a free session anymore. And so I hope you guys all sign up, because like I said, I want this to work for you, and I want this to be something that weekly we can all kind of sit down together and review, and hopefully kill the AP test and knock it out of the park. There are other things for you to subscribe to. We have a YouTube channel. There is free content on there. Not as much as the live sessions, obviously, but there are some tips and tricks videos and things on there. If you join our student Facebook page, I'm pretty active on it. I try to post at least weekly. But again, that's kind of you guys. You guys can go in there and post questions. There's other teachers that belong on that group, so there's always going to be someone there to answer your question and kind of give you advice and help you out along the way. And then if you register like you did tonight for this review session, you'll get updated emails and reminders, because normally we're on Tuesdays, but today we're on Monday. So you hopefully got that reminder because you were registered ahead of time. So other than that, that's all I really have for you guys tonight. Oops, what did I do? Nope, nothing. Okay, sorry. I really hope I see you guys all next week. And if you have any questions, I am hanging out in the Facebook group later, so you can always go in the student Facebook group and contact me there. Otherwise, hopefully I will see you next week. Bye, guys. Have a good one.